let us take the gene and its expression. Okay, result in the formation of a polypeptide. Okay, this polypeptide, it can be regulated. Okay, gene expression can be regulated at several levels. In eukaryotes, if you carefully observe, in eukaryotes, the regulation could be exerted at various levels. Okay, so at transcription level, it can be regulated. At transcription level, at a transcriptional level, formation of primary transcription. Processing level, second step is processing level, regulation of splicing, as you know very well about DNA splicing, etc. And after splicing, okay, third step will be transportation of mRNA. So mRNA transportation occurs from nucleus to cytoplasm. Nucleus to cytoplasm. Now let us learn translation level, which is a fourth step in the regulation of the gene. If you check the gene in all the cells, it is expressed to perform particular functions or set of functions. For example, if an enzyme called beta galactosidase is synthesized in E. coli bacteria, it is used to catalyze, okay, it is used to catalyze hydrolysis reactions, of particular disaccharide at lactose. Okay, so this will happen into galactose for the further coming reactions, and also into glucose. The bacteria use them as a source of energy during this process, E. coli bacteria. Hence, if bacteria do not have a lactose, imagine bacteria don't have lactose around them to be utilized for energy sources, they would no longer require the synthesis of enzyme beta galactosidase. Therefore, in simple terms, we can say that it is the metabolic and physiological or environmental conditions that regulates the gene expression. The development and differentiation of embryo, if you take the embryo, during embryonic development, the development of embryo into adult, okay, it is a complex process in the organisms, also resulted to the coordinated regulation of expressions, okay, especially for several set of genes. Whereas if you take the prokaryotes generally, they control of the rate of transcriptional initiation. Transcriptional initiation is the predominant site for controlling the gene expression. Understood, everyone? In fact, in a transcription unit, the activity of RNA polymerase, especially enzyme called RNA polymerase enzyme, at a given promoter region, okay, it is in turn regulated by interaction with the specific accessory proteins, which affects its ability to recognize the start sites. In fact, here these regulatory proteins, whatever there, can act both. Regulatory proteins can act both positively, okay, which are acting as activators, and negatively, nothing but repressors. Repressors are something repress the reaction. The accessibility of promoter, if you carefully observe, the accessibility of promoter regions of especially the prokaryotic DNA is in many cases regulated by the interaction of proteins with a specific sequence termed as operators. So operators in prokaryotes helps to process these reactions. Operator region is adjacent to the promoter region. Operator region is present near to the promoter region, promoter element. In most of the operons, so operators nothing but operons, in most cases sequence of the operator bind a repressor protein. Here each operon, if you carefully observe, has specific operator and its specific repressor. For example, lac operator is present only in the lac operon, and it interacts specifically with the lac repressor only. Now let us learn about lac repressor in the coming